So, welcome everybody to this video. So, my name is Angelika Lampert. I'm the director of the Institute of Neurophysiology at the RWTH Aachen.
And my name is Srijoy Tripathi. I'm an associate professor at the University of Toronto and a senior scientist at the Krembil Center for Neuroinformatics at KMH. And together, Angelica and I are here to present to you our latest publication upcoming in Cell.
Neuropathic pain is a burden for patients suffering from it, and about 1/3 of them is not well treated. It's hundreds of millions of people affected. In neuropathic pain, sleeping nociceptors become often spontaneously active. And we know quite well their biophysical makeup, but we don't really know their molecular targets, their makeup. And thus, it is really hard to generate new specific therapeutics for these patients.
And so can you explain what is it about these cells that calls them sleeping nociceptors? Why are they sleeping?
They are not really sleeping, so the name is really, really misleading. They are pretty active, sensing chemicals like capsaicin, pH, and other things, but they are simply not reacting to mechanical stimuli. And this is why people started to call them sleeping. Some people also call them CMI, mechanoinsensitive neural C fibers. Yeah.
And so, the problem was we know what these neurons do, but we don't know who they are at the molecular level. And without that molecular identity, it makes it really, really hard to develop new drugs that target those cells specifically.
Yeah, that's really a problem. And in the beginning of the project, I was talking with Barbara Nama, who regularly performs microneurography on humans. And we realized that we know the biophysics of these neurons really well, but not really the molecular makeup. And then there's another challenge because people who are studying these fibers, electrophysiologists, they are using a different language than the scientists who are actually studying transcriptomics and genetics of it.
That's right. And so that's where our collaboration came in. We needed to work together to build something like a Rosetta Stone to translate between these two worlds and these two different languages. We need to be able to translate between the known electrical fingerprint of sleeping nociceptors to their specific molecular signature. Like, what is the molecular name of these cell types?
Yeah, and so we started out to do experiments in the lab. We decided against mice because the sleeping nociceptors of mice cannot hardly be found in the skin and look very different. And so we went for pigs, pigs, because their skin is a little bit more similar to humans. And therefore we performed the experiments on pigs.
Right. And so, what Angelica's team did was incredibly painstaking. So, led by the amazingly talented clinician scientist, Yannis Korner, co-first author on this study, Yannis use this amazing technique called patch-seq. And so that enables, Yannis to record single neuron activity, one cell at a time. And then after he's done recording the cell, he carefully harvests the cell and then sequences its, mRNA, sequencing its genes. But this procedure is very difficult to implement, and it really requires having magic hands, and it's very, very labor intensive. And so, without Yannis's efforts, we could not have done this study.
Yeah. And then Srijoy's team, together with co-first author Derek Howard, then they collected these data. They took the data and magically made a fusion between function and the transcriptomics. And in the end, it matched. And now with this integration of this data, we had some idea, but we wanted to also see how it looks cross-species. And so we were also using data sets from humans, of course, but also other species, which were collected from collaborators such as Martin Schmelz, Jürgen Zielinski, Ted Price, and Will Rental. And then in the end, we were actually able to pinpoint down one of the cell types, which are most likely the sleeping nociceptors.
Yeah. And so as Angelica mentioned, so what emerged was this clear link between, functional characteristics of sleeping nociceptors and a single molecularly defined cell type. So we chose to name that cell type COSMR-SST after its two key molecular markers, the oncostatin M receptor, named OSMR, and the neuropeptide somatostatin. And interestingly, by sort of looking at the long list of genes expressed by these cells, we also noticed that these cells also express very high levels of interesting like ion channels. That channel and others would likely contribute to their unusual electrical properties. And interestingly, these could also be drug targets.
Yeah. But still, I mean, these were experiments in the lab and in the computer. It's not really on humans yet, right?
Yeah. So I asked Angelica, can we actually just put our predictions? Do we just test them? So, can we take our molecular markers seriously? So if OSMR really marks these cells, then could the ligand that activates OSMR, oncostatin M, you would think that if applied to the human skin, that should just specifically activate sleeping nociceptors and not other pain fibers. Is that an experiment that we could actually do in the lab?
And actually, we could. So we went back to the lab. We performed microneurography and psychophysics, testing oncostatin on the human skin, and it selectively modified sleeping nociceptors. That was actually quite a team effort, and many of us, including myself, actively supported these human validation experiments.
And this is amazing. Like, her team, Angelica's team, actually tested, my team's computational predictions in human volunteers. It almost never happens for these kinds of studies. And that really, drives home the clinical relevance to humans of what we found.
Right. Yeah. So what does it mean? Does it go forward? So for the first time now, we have in our hands the molecular identity of the sleeping nociceptors. And that, as Friedrich mentioned before, gives us these very specific targets like NaV1.9, the TTXR sodium channel, but also many other of these ion channels and other targets, downstream pathways, which offer themselves to actually now be tested.
Yeah, I mean, it's amazing, and this will be the first of hopefully many studies where we can really kind of go deep into what these cells are, and then again think about how can we actually begin to alleviate the challenge of worldwide challenge of neuropathic pain. So this whole study just illustrates what's possible when you have different groups of people working together, people who speak different languages, literally in our case German and English, and also disciplines, experimental and computational sciences, what we can do when we get together and truly collaborate.
Yeah, and this was really a transatlantic effort. About 5 countries were involved, and it was really a fantastic team. This is one of the good examples, a study which would not have been possible to perform by yourself. Not at all.
Oh, thank you. Thank you, Angelica. It was a pleasure working with you and your team and all of our many co-authors on the study. Thanks for watching, and we welcome you to read our paper to learn more about the details of our study. Thanks.
Thank you very much.